Hi, I'm Stephanie Chen, a PhD candidate at the University of New South Wales in Australia, interested in developing and optimising genomic resources for the evolutionary research and conservation of Australian plants. Today, I'll be talking about assembling the waratah reference genome. We are currently in the Anthropocene, the sixth mass extinction. The International Union for Conservation of Nature lists 27% of assessed species as endangered, with key drivers being changes in land and sea use, direct exploitation, climate change, pollution, and invasive species. Specifically, in the realm of plants, 2 in 5 plant species are threatened with extinction, and the ongoing rate of plant extinctions is up to 500 times the pre-Anthropocene background rate. Some of the major threats are shown here on the proportional tree diagram. Studying genomes can generate fundamental new knowledge of the organisation, evolution, functions, and interactions of organisms. This allows us to understand, utilise, and conserve biodiversity, and is facilitated by sequencing projects such as Earth Biogenome. Australia is home to over 21,000 plant species, and at least 84% of these are endemic. And that is part of the reason why initiatives such as Genomics for Australian Plants, which the waratah is a part of, are so important. With rapidly decreasing sequencing costs and technological advances, more and more plant genomes have been sequenced since Arabidopsis in 2000. However, sequencing efforts have concentrated on crops, their wild relatives, and model species. Therefore, the distribution of species with sequenced genomes across the plant tree of life is highly uneven. I'm working on several reference genomes. First, I'll mention the Myrtaceae species, which includes critically endangered species in decline due to myrtle rust. I've also been involved with scaffolding for a UNSW venomous snake project, and these genomes are used in bioinformatics teaching. We have Telopea speciosissima, which is part of the Genomics for Australian Plants initiative supported by Bioplatforms Australia, which aims to sequence and assemble representative Australian plant genomes across the plant tree of life to enable better conservation, utilisation, and understanding of Australia's unique plant diversity. The waratah has been the spectacular floral emblem of the state of New South Wales in Australia since 1962 and is endemic to the state. The name waratah comes from the Eora Aboriginal people, the original inhabitants of the Sydney area. It means red flowering tree. Waratah has been adopted as the name and logo for sporting teams, suburbs, trains. Here's one in the city of Sydney, and more. And a bit of etymology. So Telopea is from the Greek word telopos, which means seen from afar, and speciosissima is from Latin, meaning most showy. And you really get a sense of this if you come across a waratah out in the wild. The research I'll talk about today was published recently in Molecular Ecology Resources. Along with the reference genome, we also present 3 bioinformatic tools useful in genome assembly and curating reference genomes. You can also read about the genome in an article we wrote for the conversation. We collected waratah leaf tissue from the Blue Mountains in New South Wales. The polysaccharides proved difficult for DNA extraction, and we needed some additional and younger leaf material for the sequencing we had planned. So it didn't help that the individual went up in flames in the bushfires of 2019 to 2020. Waratah is a pyrogenic flowering species, meaning fire plays an integral part in its life cycle. So with relief, we found the resprouted individual and were able to proceed with the sequencing that was planned. Let's go through the assembly workflow. We had 2 Promethean runs, resulting in 54x coverage. NECAT produced the best nanopore assembly after exploring the assembly's NECAT, fly, and canoe, as judged by completeness, contiguity, and proximity to the estimated genome size. I did a tidy up in Diploidocus. We introduced a new method in Diploidocus for automated cleanup of genome assemblies. The program combines read depth, chemofrequencies, BUSCO genes, vector contamination, and more into a 7-part classification. Scaffolds are rated based on their classification, and one output is a tidied assembly. Next was genome polishing with Racon, Medaka, and Pylon, which included incorporating 10x linked reads to correct indels and SNPs. Then followed scaffolding, gap filling, and more polishing and tidying. You might be wondering how big the genome is. Well, it's difficult to know the true size of a genome. We present a new tool, DepthSizer, for genome size estimation using single-copy long-read depth profiles, and estimate that the waratah genome is 900 megabases, placing it between estimates by KEMA-based GenomeScope and flow cytometry. Benchmarking against several model organisms shows that the tool makes robust predictions. You can find out more about DepthSizer and Diploidocus Tidy in the paper, and the tools are also available on GitHub. Finally, Hi-C data was used to scaffold and solve the chromosome-level genome puzzle using the Juicer 3D DNA and JuiceBox pipeline, revealing 11 chromosomes before the final polish and tidy. Also, smudge plot confirmed the diploid status of the individual. Let's take a look at some genome statistics. If we consider the 10x Illumina reads, a key aspect to notice is that the genome is in lots of pieces, nearly 44,000. So why long reads? Our nanopore assembly reduced the number of contigs 120-fold, but long reads are error-prone as reflected in the lower mercury quality value, which measures base accuracy. With error correction or short read polishing and Hi-C scaffolding, we see that the contiguity has jumped up with a scaffold N50 of 69 mega base pairs. The scaffold length needed to cover 50% of the genome. Genome completeness is often measured using BUSCO, with the idea that all BUSCOs should be in the genome, universal, and only occur once, single copy. Our final assembly was 97.9% complete. It's interesting to note the difference in values between different BUSCO versions and gene predictors, here Augustus and Metayuk. The differences in those scores on the same assembly can be over 10% in some cases. We used the tool BusComp to compile BUSCO results, which can provide insight on genome completeness across different assembly steps. This tool is also available on GitHub. So now we have a reference genome. What comes next? We used Genoma, a homology-based program, to annotate over 40,000 genes, as well as R and T RNAs. The genomes have also been annotated via the NCBI pipeline. Repeats were annotated, and we found that the genome is highly repetitive, with over 62% repeats. Comparative analysis of 5 species revealed that over 23,000 orthologous clusters existed. Tests for enrichment in 912 waratah-specific clusters identified 12 significant terms, and the most enriched co-term was DNA recombination, which is interesting as waratah introgression and speciation is something I'm interested in looking at. One question with BUSCO results is, are the genes identified as duplicated a result of misassembly and sequencing issues, or are they actual duplications? We present a tool called DepthKopy to help answer such questions. Read depth analysis of 180 duplicated BUSCO genes using a new tool suggests almost all are real duplications, increasing confidence in our annotation and highlighting a possible need to revise the BUSCO set for this lineage. And that was supported also by phylogenetic analysis of all 180 genes. Copy number analysis of all the NCBI annotated genes also shows the similar clustering around the median copy number of 1. However, the mean copy number is surprisingly high, and that's been driven by a reasonably small number of very high copy number genes derived from highly collapsed repeat regions. This is the first study to quantify and kind of look at the total number of cycloidea paralogs in proteaceae based on a complete genome sequence. And those genes are key regulatory factors in the development of flower symmetry and inflorescence architecture. Our results support the idea that in proteaceae, a single duplication of CYC genes occurred prior to diversification. With a reference genome, one thing you can investigate is synteny. Here between waratah and the macadamia, the most important proteaceae crop and one of the only available genomes in the family. These species parted evolutionary ways almost 80 million years ago, and you can see an abundance of interchromosomal rearrangements, rendering it difficult to infer the ancestral form of a particular chromosome. However, for example, macadamia chromosome 11 displays a strong degree of collinearity with waratah chromosome 9, so they may have derived from the same ancestral chromosome. The Telopea speciosissima reference genome is a valuable genomic resource to understand the evolutionary origin and dynamics of the Australian flora. There is ongoing work using the reference genome together with the whole genome sequencing data, reduced representation sequencing, and chloroplast sequences to understand kind of the dynamics of all 5 species in the genus Telopea. And the reference genome gives us the opportunity to use Telopea as a model clade to understand the mechanisms of speciation. I'd like to acknowledge my co-authors who were listed on the title slide, and also the funding that has made this research possible. Thank you for listening.